Mitosis is the process where a cell duplicates itself, occurring when new cells are needed. The cell undergoes multiple phases, including interphase, prophase, prometaphase, metaphase, anaphase, telophase, and finally cytokinesis. Interphase is the first phase of mitosis, where the cell grows and starts to replicate its chromosomes. Because the cell just came out of the G2 phase, it still contains some of its features like the nuclear envelope and uncondensed DNA. It also has mitosis attributes like the two centrosomes, which are created from the two daughter centrioles. The DNA that is replicated here starts to construct into chromosomes. The next step is prophase, the longest stage, where the chromatids condense into chromosomes, replicating the two identical sister chromatids. At the same time, the nucleoli disappears. The centrosomes start to move towards cell poles and microtubules start to come out. These microtubules are called aster because it looks like a star. The elongated microtubules are called mitotic spindles. In prometaphase, the envelope fragments and microtubules extend towards the sister chromatids. The microtubules then attach themselves to the kinetochores, a region found on the sister chromatids. The non-kinetochores microtubules trap energy so it can separate the spindle poles during anaphase. In metaphase, the spindle attached to the sister chromatids align in the middle of the cell, known as the metaphase plate. The centrosomes, as you can see, are on opposite sides of each other. Anaphase is the shortest stage of mitosis. In this stage, the microtubules shorten at the centromeres and elongate the cell, ultimately separating the chromatids to chromosomes. On each side of the elongated cell, there are an equal number of chromosomes. In telophase, the elongated cell remakes the daughter nuclei and nuclear envelopes. Here the cell is genetically identical, just not yet separated. Finally, cytokinesis is where the cell cytoplasm divides and splits. This split is different in plant and animal cells. In plant cells, the cell builds a new wall, called the cell plate, between the two identical daughter nuclei. In animal cells, this cleavage pinches the cytoplasm, ultimately separating the two cells. Now we have two identical daughter cells.